time for Q&A at the end of your talks. That'd be good. I'll flag up when we've got 5 minutes to go, and then perhaps a couple of minutes at the end of each speech, at the end of each talk for a couple of questions. Any bigger questions we can tackle at the end of each session. There is time allocated. Make a note of questions if you've got them. With all events, there's a wide variety of views and opinions, so just be nice to each other. I think it's my job to keep everyone in check. I'll try my best. Fire alarms.
There's no fire alarm scheduled for today, so that if there is fire alarms that are just out the doors here.
Okay, and we're on the 11th floor, so it's stairs, I guess.
Stairs.
Yeah.
All the time.
I think Emma's been round. The event it is being recorded. We're just working on getting the live stream up and running at the moment, so it'll be on YouTube. I don't think the camera. I think the camera can only see my face at the moment for all those poor people on the internet. If you do have a question, if you want to remain anonymous, don't say your name. At the same time, if you do want people to know who you are, then please feel free to tell us who you are, and it will, be captured. No swearing. We will be taking some photos for publicity. This is the 3rd symposium that we've run in this space, I think in 24 months, the first UK paediatric symposium, so I think it's important that we can talk about this. And I think Khadijah's got a feedback form. That's right. Excellent. So I'd be really grateful if you could complete that towards the end of the day, because it means that we can make these events bigger and better in the future. We will try to keep on time. We aim to finish no later than 4:30. There are breaks at 11 and 2:45 with lunch at 12:45. I think that's me done. Have I missed anything on the sort of pragmatic side of things? That's it. Thank you. So without further ado, I'd like to introduce our first speaker. It's David Walker, who is Professor of Paediatric Oncology in the Children's Brain Tumour Research Centre at the University of Nottingham. David's going to be talking to us about recent research and the drug delivery challenge in paediatric brain tumours. Thank you.
Thanks very much indeed. Now, am I going to use that, or Thank you very much. I've known some of the people in the room. Which some of the researchers and some of the families and patients, and it's nice to be here. If I could have my first slide, it suddenly disappeared. Modern medicine, you have to be an expert in all things. Yeah. So I have worked in Nottingham as a clinical academic, consultant and researcher since 1990, and we set up the Children's Brain Tumour Research Centre in 1997 with a fundraising campaign through the University of Nottingham. And that started to bring money in, and some of the people in this room benefited from being employed in that money in the early part of their career, and Beth, you're going to hear about later, who was our first academic appointment, so it's a great celebration to see her here today. The Children's Brain Tumour Drug Delivery Consortium, which is Emma Campbell here, and I co-lead it with Ruman Rahman, was set up to promote awareness of drug delivery as a way of enhancing treatment for the future in brain tumours. And this is part of the vision of the Children's Brain Tumour Research Centres. We want to create a situation where the children who get experience brain tumours can achieve a full, healthy and independent life for every child. And we wish to do that through research and accelerate progress. And so that's our mission. And that picture there is of the Vice-Chancellor of the University in 2014 and the children for whom we cycled the country for 1,400 miles to raise £2.2 million in that year for brain tumour research. So we had very, very substantial backing from the university leadership for this programme. These photographs are for the second sponsored event that took place in 2017. again, to raise money for brain tumour research. And we now are a named charity for the university sports teams, and so all the sports teams have brain tumour research on their T-shirts when they play sport. So this university has backed this topic maximally in a really very gratifying way. So I'm going to talk a little bit about drug delivery, and this slide summarizes, points out the website delivery consortium here, and if you're welcome to visit that, there's quite a lot of information, and we're trying to encourage researchers, regulators, the drug industry, strategic investors and research to say that if we want to solve the problem of brain tumours, we not only have to think about which drug might work, we also have to think about how do you get it to the tumour. And the bottom line is that the brain and the bloodstream are divided by something called the blood-brain barrier, which physiologically prevents drugs or molecules, large molecules, water-soluble molecules, from penetrating the brain. And this means that the drugs that are produced by the drug industry, unless they have characteristics designed to cut across the blood-brain barrier, don't get into the brain, and probably don't get into the brain tumours as a result. And so our strategy is to say, yes, we need new drugs. Yes, they need to be targeted against molecular steps in the cancer development pathway. But more importantly, they need, in addition, to be got into the tumour with a specific mechanism. And so that's what I'm really going to talk about today. And we identified this as a priority in thinking about the different approaches to enhance. I'm not quite sure why that keeps coming and going. And so we talk about biotargets, and there's an enormous amount of work has been done looking at biotargets. And we know a lot about the molecular environment of the tumour now. And there are many drugs that are candidates for tackling those targets. But at the moment, most of the drugs that come out of the drug industry do not penetrate the blood-brain barrier. And so our hope of finding a target and a drug may be compromised by this pass through the blood-brain barrier. We're also interested in brain injury, and some of the people we'll be talking about later in the day. So, drug delivery is our theme, and there are different ways of doing this. And this is an example. One of our researchers developed this cartoon, and it shows the idea that you have a cavity in the tumour, and you may put a bioadhesive gel into the cavity, which contains drug, which then delivers the drug to the cavity. That's a simple concept of drug delivery. But how you make that safe in clinical practice is complex, and there are people in the room who can talk about that later. So today, what I want to do is introduce the concept of drug delivery. I started that. Think a little bit about the blood-brain barrier. We've mentioned that. Want to think about reasons for drug resistance. So why do some tumours not respond to drugs, and why do others? So that's one thing. And then talk a little bit about drug delivery systems that are in development, and I'll show you a couple of examples. And then at the end, I hope to show you something from the patient's perspective. Everybody talks about brain tumours, but actually the pathology have just reissued their classification, and there are over 110 different types of brain tumour as identified under the microscope. So that's actually quite bewildering, because if you get very precise, that means you have 110 different unique targets in one, and that's with just one target per tumour. But each, when you come to study these tumours, there are often 5 or 10 targets that are potential. And so you have a very large number of biological targets potentially when you talk about brain tumour. In childhood, there are a subgroup which present predominantly in young people, and I'm going to very briefly tell you which they are, because it helps you understand that no one problem, there's not a single problem here. So the top left here is a piloathletic astrocytoma. This is a benign brain tumour, and the surgeons are very good at removing this, and they almost never need any further therapy, and 90% of them are cured. The problem is, and these marks mean this, that they can have ataxia, unsteadiness afterwards, because the cerebellum's the coordination center. And also they can, if they've had bad delay in diagnosis, they often lose vision. So they can have impaired vision. This one here is a hypothalamic astrocytoma, which is a tumor in the middle of the brain and the hypothalamus that controls your appetite and endocrine system. And they often present in very early childhood, in the first 2 years of life, and they have funny eye movements, and they often don't grow properly, and so they have problems with nutrition, because their metabolism's working too fast, and you can't give them enough food to fill them up. And so when we treat those, about half of them respond to chemotherapy, but half of them don't. And so they sometimes do die, sometimes they respond very nicely. So they're a potential target for drug delivery because not all of them respond to therapy. There are new drugs coming along which may be suitable, but they suffer from blindness and endocrine problems after they're treated. This is a group of tumors linked to a condition called neurofibromatosis type 1. And that's a genetic disposition syndrome, and can present tumors in the optic path and prevent blindness. And in fact, the treatment there is not to save their life, but to save their vision. So blindness is a problem, and they also have endocrine problems. And so, yeah, try that. So they are probably not suitable for drug delivery, although we are getting a whole new bunch of drugs to test these, and we're assuming, we don't know, that the drugs will cross the blood-brain barrier. They're about to launch a trial, but they don't know for sure that they cross the blood-brain barrier. Brainstem glioma, this is a low-grade brainstem. There's a tumor here right at the top of the brainstem, and that's where all the main nerves from the brain go down to the rest of the body here. This is a low-grade tumor, and Stuart, who's our neurosurgeon, if I asked him to operate on that, he'd say, no, thank you very much, I'm likely to paralyze the patient. So he can't take the tumor out. So what can we do? Well, we can treat these with drugs, and we can treat them with radiotherapy. It's very central placed, and they sometimes respond to drugs, but they don't always. Normally they respond to radiotherapy, but the risk is that they end up with paralysis from here downwards, because of where it is. Similarly here with a spinal astrocytoma, if I ask Stuart to operate on that, or one of his colleagues, they'll say, oh yes, sometimes we can get these out very successfully, but sometimes there's no plane, and so we can't get them out. And then you have an option of either doing nothing or giving some trial of drugs or some radiotherapy. They can be problematic, and they can paralyze the patient. These two groups of tumors are medulloblastoma, and Beth's going to talk about those. They're malignant brain tumors. They occur in the back of the head and the cerebellum. They can spread down the spinal cord and around the brain. And if they present under the age of 5 or 3, the brain's still growing and developing, and so it means if we use radiotherapy, we'll damage their memory and their growth very severely, and we tend not to use it. And so they're a group of patients who, because they can spread, they're wondering whether we should deliver drugs to the spinal fluid directly as a way of drug delivery, because that may be a better way of treating them and giving very big doses of chemotherapy or radiotherapy. And if they happen over the age of 5 or 3 or 5, then we can use radiotherapy, and something like 80% of them are cured as long as they don't have metastases. This is a patient's tumor that happens in adolescence, and particularly it's called a germ cell tumor. And they present with hormone problems, so they often have abnormal puberty, and they may be blind when they're presented because they get hydrocephalus, and they can spread down the spine. They're actually very sensitive to chemotherapy and radiotherapy, and 90% of them are cured. And they may be a group of tumors where if we deliver drugs to the spinal fluid that could save us irradiating the whole of their spine, but it hasn't happened yet, but it's something we're thinking about. This is a tumor that our centre are particularly interested in, ependymoma. They occur in the back of the head, mainly in children, and they can occur in other parts of the brain in adults. And they're very large often when they present like this done. And the surgeon has really got most of the responsibility for curing these. If they can get them all out, then there's a chance of being cured. If they can't get them all out, then generally they keep coming back, and drugs are almost universally ineffective at the moment. And it may be that delivering drugs directly to the tumor maybe is an area that we haven't explored yet, or delivering, and Stuart might talk about this, electric fields in that area may be a futuristic way of approaching these. And so we're really interested to know why they're resistant to chemotherapy, and it's not clear. So that's still work that's undergoing. This is the signature tumour that we worry about drug delivery, and I'll tell you a little bit more about that. That's what they call a diffuse pontine glioma. And some of you in this room may know children or have had children who've had this, and it's a very distressing condition. It's the one, when we meet somebody whose child's got this, the current evidence says that 90% of the children are dead in 2 years, and there's no effective therapy apart from radiotherapy, which is temporarily effective, and then the tumour regrows. And it is an area, these are high-grade gliomas. They have now got a new molecular signature that tells us that they come from a certain developmental pathway in the brainstem that makes them attractive, and maybe new targeted drugs will come forward. But this part of the brain, the blood-brain barrier, is particularly tight. And so we give the drugs into the bloodstream, they probably won't get in. And so this is an area I'll talk about later, where we're using a technique convection-enhanced drug delivery, where we're actually injecting drugs directly into the tumour to try and bypass the blood-brain barrier, and I'll show you evidence of that. And then this one here is a atypical rhabdoid tumour, and these occur in very young children. They're very aggressive, very fast-growing, and you have to use very intensive chemotherapy with both into the bloodstream and into the spinal fluid to try and control them. And we are making some progress. There is some improvement, and we also understand the biology of this disease, and there is a possibility. But drug delivery is already part of these treatment programs with intra-CSF therapy, and there may well be other treatments in the future. So that's a whistle-stop tour of the whole of neuro-oncology in about 5 minutes. That's taken 30 years to get to that point that I can do that and not be challenged by everybody saying, I'm not talking about it, I'm not sure what I'm talking about. This is a word map that I asked an experienced audience in London to produce at a conference, and asked them what they meant by what meant to them precision medicine. And you can see that the biggest word was targeted. And I think in the minds of the people there, they were thinking of biologically targeted, actually finding the marker and the drug combination. But actually, in brain tumours, I think targeting the drug to the tumour is another really important part of precision medicine, and that's what the Drug Delivery Consortium is seeking to enhance in people's minds, is that we don't think about how we get it to the tumour, we may not get our drugs to work. So we developed a concept here with our collaborators of hatch, match, dispatch, and deliver. As a kind of racy title, is how do you get a drug to market? And you had to hatch an idea, so you had to think about the biology of the drug, of the tumour, and you think about drugs that might tackle that biology, and you might say, well, that's the first thing. And then you had to think about how you might combine these with a drug delivery system. So that was matching them. And then you'd have to do some research to prepare them for clinical practice. So that's doing some early research in the laboratory to say this drug works, this delivery system works, it's safe to think about putting it into patients. And then you have to deliver it in a clinical trial. And we're going to show you 2 examples of where we've tried to do this. Hatch, match, dispatch, and deliver. And this is the first one. I'm working at the moment with a group in Bristol. Seems to have a life of its own, this thing. A guy called Steve Gill. He's a professor of neurosurgery in Bristol, and we're working with, several of us are working with his team. And he has developed a number of pieces of technology working with a company called Renishaw. And the first piece of technology is this catheter. This is a micro catheter that can be put into the brain. And the unique thing is it's got a little sleeve on the outside that prevents reflux of the drug up the track that the catheter goes through the brain. And this delivers a globular convection-enhanced drug distribution around the tip of the catheter, rather than it refluxing up the catheter side and just coming out the surface of the brain, because that's what happens if you don't have that cuff. The second thing he developed was a bone-anchored device through which he could anchor these catheters when he placed them into the tumour directly with a robot, driving the catheter into the precise location that he wanted to put the catheter tip. He can then connect them all up to a single catheter, which he then brings out on the surface of the skull with a syringe hub here. And that's a picture of the child syringe hub, to which we can attach an infusion device so we can infuse drugs directly into the brain. And this is a video I'm going to show in a minute of that happening to that little boy. And these are 2 videos here. So you just click. Yeah, go back. No, sorry. Click on that one. Yeah. So this shows you, if you can see it, this is a movie MRI scan showing the tumor in red in the middle and the 4 catheters going directly into the tumor, 2 from the front of the skull and 2 from the back, so that the catheters go directly into the tumor. Just press that again, just let people see it again. Keep it moving. And then if you go over to the one in the bottom left and run that one, it should be a video. And this is a silent video. There's the catheter externalized, and this is the process for connecting it. And what's really interesting about this is it doesn't hurt, which for children is really important. And this is the operator linking up the infusion. We can run 4 infusions at once into the cat at different rates if we choose. And the aim is to encompass the whole tumor in drug infusate, so that the drug is actually soaking the tumor bed in its entirety. And he's connected, use a bit of Meccano, you have to use a screwdriver. You connect the circular device onto the hub, you then screw it tight to make it tight, and that's what he's now winding the needles into the hub, which connect up with those 4 catheters I showed you on the device, so that we know which syringe is delivering to which catheter, and we have a catheter map knowing exactly where the drug's going. And then this is the boy having his infusion. The infusions go on for about 12 to 16 hours, and they have play therapy, they watch telly, they do their homework, they talk to their friends and family on Skype. They're perfectly well. They can have fluctuations in their neurology, so we have a scoring system that measures how well their brain's working, and the nurses apply that on an hourly basis during the infusions, and we occasionally see some of the nerves functioning less well. We slow the infusion up, and that allows it to recover, and then we restart the infusion. So that's a way of delivering drug directly to the tumor. It's happening now, and we hope to launch the trial in next year for patients to be recruited to give drugs into that route. And the drugs we've chosen to use are carboplatin, which is a chemotherapy agent that we've all used for many years, which if you're given doses sufficient to penetrate the brain causes severe bone marrow toxicity. But in the quantities we use here, it has no effect on the bone marrow whatsoever. The other drug we're combining with is sodium valproate, which is an anti-epileptic drug, which also attacks the HDAC molecular mechanism in the cell and arrests tumor progression with that, and the 2 things work synergistically. And so we're combining these 2 drugs to try and see if that will produce a better response. We have seen responses in patients treated on compassionate use, and we have seen people live much longer than we predicted as a consequence. And the reason that this is a particularly important model is that a tumor of the brainstem is in the part of the brain that keeps us all breathing and our heart rate going. And so if the tumor progresses there and causes damage, then we tend to die from breathing problems and heart rate problems. So if we can control the tumor in the brainstem, we would expect to keep the patients alive longer, which would give for us longer to think about other ways of treating the rest of the problem of this tumor, because they do spread beyond the brainstem. And so we need to think about how we're tackling that. But we have to get step 1 right, to show it's safe to do this in the brainstem with this drug combination. So that trial has currently been moved to beyond the first stage of approval with the national groups. The European Medicines Agency have approved it, so we put it for ethics approval at the end of this month, and hopefully open it in March of this year in London. All the patients will be treated at Harley Street Children's Hospital, free of charge. There'll be no charge. The hospital's bearing the cost of that, and we're seeking other funds. So that will be done in that particular hospital environment that's been designed to accommodate that needs for anybody, and it will be an international trial, so people can come from outside the country, can also come from the UK. So to get to that point, the Bristol team had to do the preparation for dispatch, which is the molecular work to say that the tumor would be responsive to these drugs, that the drug combinations would work, They had to use animal testing, and some people may have sensitivities about that, but we can't do this sort of thing in humans without testing it in animals first. And so all that was done in order to get us to the point that we are at now. Next. And the challenges of this are summarized here. We're working in new ground. How do we know how much drug we're giving? Well, we know how much is in the syringe at the beginning and how much is in the end, but we don't know how well it's distributed. So finding ways of looking at the drug distribution using MRI is an area that we're looking at. The MRI is a fantastic technique, but how we actually measure the distribution of drug is really important. Measuring the response. These tumors fill up a structure, and they don't always shrink when they respond. They just don't progress. Sometimes they swell before they shrink, and so that is a problem in our assessing it. It's opening the door to repurposing drugs. I said at the beginning that the drug industry make drugs that are water soluble and not fat soluble, and that means that most of them don't get into the brain. This is a really good thing for this technique, because actually water-soluble drugs don't get out of the brain. So if you put them directly into the brain, they don't get out very well. So it's opening the door to a load of drugs that were rejected, probably because they didn't get into the brain, and may well open it up for new use in this way. But it's a small number of patients, it's going to be slow progress, and it's got to be safe. We can't make the children worse with this. That's really important. And how do we do clinical trials? This is a new technique, there are new things, and it's been very challenging, and our colleagues have provided with loads of guidance, and we're now getting to the point where I think the trial is acceptable, but there was a long period of time where there was a lot of criticism, and I think we've addressed all of those criticisms. We are moving forward. Okay, move on. So, move on to the next one. Don't worry about that, we've seen that already. So, the second area that we've particularly taken interest in is, and I mentioned it earlier on, is giving drugs into the spinal fluid. Now, the brain is floating in spinal fluid, and in leukemia therapy, we've been giving drugs into the spinal fluid for 30 years. And it's been shown to be a very effective way of preventing CNS recurrence in leukemia. But it hasn't been used extensively in brain tumors. And I'm not really sure why. There is a video which we haven't got time to watch now, but we can send that out to all the participants so you can see it later. But this is a diagram to show the places where we can give drugs into CF. We can either give it into the ventricular system in the brain, or we can give it into the lumbar root down here. And we preferentially give it up here because the CSF is made up here and flows down the spinal cord and is reabsorbed in the outside of the brain. But sometimes there's an obstruction up here due to the tumor, in which case we give it down here and then they travel up the other way. So we've got a way of dealing with anybody who has a brain tumor if we talk about intra-CSF therapy. Next slide. So we did some early work, and we looked at which drugs would be suitable for giving into the spinal fluid, because the spinal fluid is in direct contact with the brain. If you have a drug that's got very toxic properties, you don't want to give it directly into the brain. It can be devastating. And so we selected a drug called etoposide, which is well known to us as oncologists, and we gave a patient oral etoposide every day for 2 or 3 weeks. And we also gave the same patient CSF etoposide. And we saw response in the and around the CSF spaces, but progression in the tumor, which gave us confidence that the method of drug delivery was important for the effectiveness of this drug. Next slide, please. So if we direct drugs directly to the CNS, you can achieve cytotoxic tumor concentrations bypassing the blood-brain barrier, which saves a lot of drug and toxicity. But you need to match the effective drug, the biology, and the optimal delivery system. Next slide. So Michael Vlubergs, who's a neurosurgeon, works in Nottingham, did a lot of work on understanding the flow of CSF around the brain and spine. And he made physical models, he made mathematical and computer models, and he studied the flow of CSF, because if you're going to inject drug, then you need to know where the drug is distributed. So we've developed an enhanced understanding of that using various engineering principles. We worked with a man who specialized in getting coal into power stations. He said CSF was just coal in smaller lumps. He was an engineer. Next on. Next slide. And I'm not going to go into that now because we haven't got time. And just the next slide. So we've had some preliminary experience of using intra-CSF drug delivery in children with relapsed brain tumours. And this slide was taken from a poster presented in CEOP in Japan 2 weeks ago. So we've treated over 25 patients with intra-CSF etoposide as part of their palliative therapy for brain tumour after it's recurred. And there were 16 male and 13 female. The median age, that half of them were younger than 4, the other half were older than 4. And the sorts of tumours we treated were medulloblastoma, ependymoma, pineoblastoma, lymphatic leukemia, can't read that, PNET, and a number of other ones. So it's a variety of patients. And we got this survival curve. Now, sadly, they all had relapsed disease, and so they were all at risk of dying. The red line here is the predicted median survival, and the yellow line is the actual median survival. So we think, first of all, we were able to treat them for an average of 325 days, which is nearly a year, and we think we prolonged their progression, their survival as a result of this. And if you watch the film when you have more time, you'll see the patient saying how tolerable this was and how it really allowed them to get on with their lives without having lots of disturbance, and nothing like as bad as it was to having systemic chemotherapy. And so we concluded from that we can give intraventricular etoposide. It's well tolerated. We had quite a lot of problem with infection of the device. 24% of them had to stop the treatment because the device got infected. And so this remains a problem. And Steve Gill, who designed the other system, we are thinking about whether we can make a device that might bypass the problem of infection with this idea of having something that comes through the skin. So we're thinking about that. Next slide. We designed a trial called Intrepid, and we got funding for the trial from CI UK to study intra-CSF drug therapy. And we're all ready to go, except for one problem, is we haven't got a reliable system for attaching a semi-permanent infusion to the device in a safe way. And we had thought we could run a catheter from the scalp under the skin and put a portacath here, which is the device we use for intravenous administration. But we were told from a governance point of view, we couldn't have one going to the venous system and one going to the brain, because there could be a risk of error of people giving the wrong drug via the wrong route. And so the trial is currently suspended until we find a solution to that practical problem. So progress in research as rushes forward and then meets blocks, and then you have to wait and sort something out and then rush forward. And I think we're prospect of creating a device that could be used which avoids the portacath solution is in our minds, but it's got to be created, and that would allow the trial to go ahead. Next slide. So that will bring me to the end. So the idea of hatch, match, dispatch, and deliver, I've tried to talk to you about how you take practically a translational research journey from an idea through to an end. And I think we've got now one trial about to be launched. We have a second trial designed, but waiting for that last delivery element to come through. And then there are a whole raft of other things to do with drug delivery, which I'm not going to talk about, which could well be developing in parallel. So we feel that the opportunities going forward are quite good in this area for research and investment and for realization of benefits. Thank you very much.
David, thank you very much indeed for your time on that. Quite a journey from what started off as being, for me, fairly straightforward sort of concepts, and the massive impact of the disease across 110 different types of tumour. I mean, the massive granularity of the challenge is just, it's terrifying, frankly, from a research perspective, I imagine. I think, lots of questions, but let's, in the interest of time, if you don't mind, if we could take, we've got a 10-minute sort of window at the end for questions, I think, from the 3 speakers we've got in this first session. So suggest, if you make a note of your questions, we'll pick them up shortly. I'd like to welcome Beth Coyle to the, I was going to say stage, to the floor now. Beth is Associate Professor of Paediatric Oncology at the University of Nottingham. And if she can find her slides, she will be talking to us about modelling medulloblastoma metastasis. It's on the big screen. Here we go.
I'm probably going to start trying to click this. Okay, so I'm going to take you now to kind of lab-based research, and hopefully I've pitched this at a level that'll keep you all with me by the end of my talk. So here we go. So a lot of my research has focused on medulloblastoma. This is the most common malignant pediatric brain tumour. It usually occurs in children. The peak age of incidence is between the ages of 3 and 8. It occurs in the cerebellum, as David's already mentioned, at the back of the brain. But the big problem with medulloblastoma is at diagnosis, a third of patients are already metastatic. This means that the tumour has spread through the CSF. You can see the arrows and the flow of the CSF on this picture, or through the ventricular system. We're still developing our understanding of that. But where it ends up is either forming laminar, so a sugar coating across the top of the brain or down the spine, or it forms solid mets. And these are called nodular metastasis. The problem with metastatic disease, which David's already alluded to, is it responds very poorly to current therapies. Patients with metastasis or tumour spread are given more aggressive treatment. So they're given high-intensity chemotherapy, and if they're over the age of 3, they're given even more radiotherapy. And the consequences of this are pretty devastating. It damages the surrounding brain, it affects their hormones and their IQ, and it causes peripheral nerve damage. So what we need are biologically relevant treatments. And through this, we hope that we will increase survival and also, importantly, increase the quality of life for survivors. So what do we know about the biology of the disease for medulloblastoma? So I'm not going to go into the types of molecular analyses that have been done, but a great deal of molecular analysis has been done. And the last 8 years, it's become much clearer. And medulloblastoma splits into at least 4 different subgroups. Within those, it becomes more complex, and there are other subtypes, but mainly there are these 4 clinical groups. And these are highly clinically relevant. I don't know which one I can point to. If you look at the top blue line, this is the Wnt subgroup. Under current therapy, these are essentially curable. However, you have sonic hedgehog in group 4 in the middle, and this is group 3. And the survival rates are very low for this molecular subgroup. But when we think about metastasis, actually it maps directly onto it. So what we see is in the Wnt subgroup, you don't tend to see metastasis. You will see it occasionally, but not very often. Sonic hedgehog, one of your intermediates, you see a low level of metastasis. The highest level is in this group 3, with the group 4 coming close behind it. So we know there are clear molecular differences indicating that the drivers of the tumour are different for each of these subgroups. Now, traditionally, if we're going to test drugs, what's been done is they've been tested initially in the lab in 2-dimensional culture. You take the patient's cells, you develop them so they'll grow as a cell line, and we grow them across the bottom of a flask in a solution that's physiologically matched to the tumour. The next stage of this is to take your cells into a mouse model. So you'll take the tumour cells and implant them, either as shown here in the brain of the mouse, and then if we label them, we can follow the cells as they migrate and metastasize down the spine. And this is one of my models in the lab. We can also do genetic manipulation. If we're really keen and we see a pathway, it's really important, we can make a genetically modified mouse. And so we alter it and we can produce a colony of mice that will always have that defect that you see in the tumour, and you can use it for testing drugs. However, based on these types of studies, the success rate for new drugs in phase 1 clinical trials is still about 3%. And of the drugs that make it through the phase 1, only 1 in 10 of those drugs will progress to give patient benefit. And none of the drugs that are used to date were developed specifically for children. So they're not built on the biology of the tumour. So the aim of my research group is to develop new, more representative models. And we're doing this by building 3D culture models. And what I mean by three-dimensional is that we grow balls of tumour cells. So instead of growing them flat across the bottom of the plate, we can grow a sphere of cells. So it's three-dimensional, it's much more representative of the tumour. We can also grow these on or within matrices, either a simple matrix, so where we're modelling things like the blood-brain barrier or basement membranes, but we can also tailor and build more complex matrices, and I'm going to tell you a bit about that. The important thing about these is that high throughput. We can either do them in 96 well plates or 384 well plates, as I've shown you here. This plate is the size of my hand. Each well that we grow our cells in are the size of your pinky. So in one plate, we can carry out experiments that equate to 384 mice. So we can get a huge amount of data in our representative models. It's far cheaper than using mice, and also the material that we put in is humanized. So we can model things that are much more relevant to humans. So the first that I'm going to tell you about a little bit more depth, this is being worked out by Sophie Roper, who's a PhD student in my group. And she's built some of these spheroid models. So what we do is we take our single cells, we allow them to form our spheroids just by stopping the cells from sitting down. The plate is a low attachment, so they can't sit on it. So they form a ball naturally. Sometimes we need to give them a few other factors to help this to work. And we've also realized that we need certain growth factors in there, but we've worked out that this works across all subgroups of medulloblastoma, although some of them form a little bit looser spheroids. We form our spheroids by day 4. By this point, the diameter of them is the maximum diameter to allow perfusion of drugs. So we can study drug penetration into these. We can also look at their growth, drug responses I've mentioned, and we can take them further and look at migration and invasion. When we look at drug response, if you look at these, we've got a grey line and then a black line. And the blue arrow that I've put on shows a distinct shift in drug response. So these tumours, when we grow them in 3 dimensions, are much more resistant to therapies. And if you look at the one on the right, you'll see actually we have a survival of a drug-resistant population. We don't see that in 2-dimensional culture. So we're starting to develop something that's much more clinically relevant. And we can image these as well. If you look at the one on the bottom right, you'll see that this ball of cells at the end is very resistant to chemotherapy. Whereas the ones on the left, where we see a response, they're falling apart in response to chemotherapy. So we can start to model this within our system. When we think about migration, we can take our cells, if I can get this to work. Is it gonna work? Yes. Have you got it running on the other ones? Okay. So what we see here is the cells being placed on a basement membrane to mimic some of the barriers in the brain into the CSF or into the blood system. And you can see the cells start to migrate out and join together. So we can start to inhibit this process. What we also observed was that there were little tiny particles coming out ahead of the cells. So if you look more closely, I don't know how well you can see it on this, but there's some little dots there forming ahead of them. We were interested in what these were. So we think that these particles that are being released by the cells are these little blue membrane-bound particles that I'm showing you here. And these are called exosomes, and they're carrion material, and they're running ahead of the cancer cells, and they're signaling. And now exosomes are very common throughout the body. Most cell types will produce them, and they're found in all bodily fluids, which includes the CSF. And they have many important functions. But within cancer, we believe that these exosomes are actually transferring important messages. We think that they're going ahead, they're promoting tumour growth, they're enhancing migration invasion, they're promoting therapy resistance, and they're also dampening down the immune response. So they're helping the tumour to grow. And this is an image showing some of the exosomes being released by our cells, these little green dots. So in medulloblastoma, we think that what happens is the tumour releases the exosomes, and these go ahead and prepare the metastatic site. And our proposal is to look at these. Initially, we're modelling them in cell culture, but we believe if we take them and we extract CSF from patients, we'll be able to isolate them and use that for early diagnosis and therapy selection. And also, we can use it for therapy monitoring. Alongside this, I've got another PhD student, Christine Matoko, who's a neurosurgeon, and she's looking to develop MRI imaging to detect these within the CSF, so we can use that prognostically and for therapy monitoring. The more advanced models that we're developing are our brain-inspired hydrogels. So here, what we're doing is modelling the microenvironment of the tumour. So not just a ball of tumour cells, we're allowing the tumours to develop in a matrix that is matched to the microenvironment of the brain where they grow. We're using the backbone of the brain initially, and then we're altering that to fit with the different components found in the different subgroups. And through this, we can look at cancer growth, we can look at movement, we can look at response to drug treatment, and also we can put immune cells into these models and build much more complex models. This should start right away. So this is one of our tumours growing within the matrix. And here you can actually see the full migration profile of the cells. So we can monitor that down the microscope, and we can also look at response of these down the microscope. So the final stage that I'm going to tell you about today is Masha's project, and Masha's here in the audience. And Masha has been looking at how the cells move from the tumour into the CSF. And she's been building a blood-brain barrier model so we can look at the migration of these purple cells through. And what we're going to do is combine that with our hydrogel mini brain models. And so then we can put together a brain tumour with the CSF, and we can model that migration. We can look at putting drugs into the system, and we can look at inhibiting it. So that's my kind of whistle-stop tour of what we do to model medulloblastoma. Just to put it in context, so I've told you kind of about, our work is very circular, and I've told you about the middle bit of it. We also do some work where we're developing prognostic markers, so we have some work that's candidate-based, where we have some hypotheses of things that we think might be involved in this process, and this is being undertaken by Alice, who's here in the audience as well, and Louisa. They've got some markers that we've shown to be prognostically relevant, and they are testing these within the model systems. We're also doing convection-enhanced delivery work, where one of the drugs that we tested in our system, which was highly effective, we've taken forward into preclinical work where we're looking at CED delivery. And we actually switched that to diffuse intrinsic pontine glioma, because that was a better system. And we showed the same drug, which is a novel temozolomide analog, to be just as effective in DIPG, and we're working with that, and hopefully we'll bring it back to medulloblastoma. And finally, just to say thanks to everyone that's involved and our funders. Thank you. Did you hear me? I didn't really use it, did I?
I could hear you.
Sorry.
Thank you very much, Beth. It's fascinating. Next up, we have two speakers, I believe. Is that correct? You both here. It's Dr. Raman Raman and PhD student Catherine Vasey. Is Catherine joining you?
I'll speak for the first 10 minutes.
Okay, brilliant. Thank you. And I understand you'll be talking about delivering drugs during surgery. Wonderful.
Should be. Put it on a stick. Yeah. Okay. Okay, good morning everybody, and thank you Brain Trust for this kind invite for many of us from the Children's Brain Tumour Research Centre at the University of Nottingham. And this talk that's entitled Delivering Drugs During Surgery is going to be divided into two parts. I'll speak for about 10 minutes on an existing technology we've been developing, and then I'm going to hand over to Catherine Vasey, who is a second year PhD student working in my group, who's going to take you into the world of the very, very tiny and tell you about some innovative nanotherapy which is building on this existing technology. So as David alluded to in the first talk, there are some fundamental problems delivering chemotherapy systemically. And when I say systemically, I mean delivering drugs through either the bloodstream or patients taking drugs orally. And one of the problems is that most of the drugs administered in this manner does not reach the target site, which is the malignant brain cancer. And due to the spread of these toxic agents throughout the body, patients often experience a lot of toxicity to healthy tissues and organs, and this further confounds the ability to deliver therapy doses to the brain. So because of these toxicities, the dose that would be effective in the brain is often not achieved. And finally, not least, there's a physical barrier called the blood-brain barrier. And this is, in healthy individuals, this is a good thing. We've evolved to have this barrier which protects our brains from infections such as bacterial toxins and pathogens. But in the case of delivering chemotherapy drugs, they fail to get into the brain because of this poor penetration. So with that in mind, there is a huge interest that has been for almost 2 decades of delivering drugs locally at the site of brain cancers. And this is the rationale for this is that the application of chemotherapy during surgery or immediately after surgery, directly in close proximity to the cancer that cannot be safely removed and is left behind, presents a window of opportunity and a unique manner of delivering chemotherapy. It's been demonstrated before successfully. Here's an image of a high-grade brain tumor on the left in the MRI scan. And this strange, large polymer you can see here, this is a material that is a solid wafer called gliadel wafers, and it contains a drug called carmustine. It was developed in the late 1990s in the US. And it's, as you can see here, the neurosurgeon places about half a dozen of these into the cavity in close contact with the cancer cells that have been left behind. And this drug carmustine is then seeps out from these wafers. So the main point here I'll stress is that this is a preformed solid material. So although it is placed in the cavity, it doesn't necessarily have very, very close contact against the cavity lining. So the rationale for local delivery in this manner is the potential to achieve high local concentrations of the drug, but at the same time you're minimizing or even completely restricting chemotherapy drugs throughout the systemic delivery throughout the system. And this is the clinical data. This gliadel wafer delivering carmustine has been approved for use in the clinic. It's not widely used. It's given to some patients who do respond. And you can see here from the survival curve, the group that received carmustine has a small, moderate 2-month survival benefit. So this was the impetus for our project in the University of Nottingham. And we developed a chemotherapeutic paste, which I'll call PLGA-PEG. It is a polymer, but the next speaker is going to be far better equipped to tell you exactly what a polymer is. But here are some of the advantages that we felt we had over the gliadel wafers. So this PLGA-PEG paste is biodegradable, which means that when it breaks down, the components of it are safely removed by the body. The idea that this can be pasted around the cavity wall is in contrast to that solid, already preformed wafer you saw in gliadel. We can also deliver multiple drugs in combination, and ultimately we think there is potential advantages over gliadel because of all these factors. So I'll first show a schematic cartoon, and then I'll show you a video of this in action. So PLGA-PEG are microparticles in terms of a scale of size. If you think of a diameter of 1 meter, so a small desk, this is about 1 million times smaller. So these individual particles are 1 million times smaller than a meter. So the PLGA-PEG microparticles, when mixed with water containing chemotherapy, forms a paste, which is what attracted us to this material, that this can be applied to the very irregular shaped surgical cavity wall. And due to a very neat property of the material, it solidifies only at patient's body temperature, and it does so within about 10-15 minutes. And then ultimately, when it's held in place in that way, the idea is that it will deliver the chemotherapy drugs, and hopefully sufficient amounts will reach deep into the brain where we know there are cancer cells behind. So there's going to be a few videos, I think, particularly in Dr. Smith's talk, showing the brain. So this on the left is just showing, this is a sheep cadaver brain, and this is showing the ease of application of this paste. So this was really critical for us to show that this is something that wouldn't be too laborious to use and wouldn't require much training by the operating neurosurgeon, and could be applied relatively quickly. So you can see here, this is a cavity in the brain that's mimicking a tumor cavity, and the neurosurgeon is applying this PLGA-PEG containing chemotherapy as a paste. And then the video on the right is the same. It's the same cavity after about 37 degrees for about 10 minutes. And you can see the paste is now conformed and taken the shape of the cavity. So the difference between this and gliadel, our hope was that this is in far closer proximity to where the remaining cancer cells are. So when the chemotherapy drugs come out, they have a little bit more opportunity to diffuse into the brain. Okay. And then in the early stages of the project, it was important that we showed that there would be realistic clinical utility with this material. So we did a few tests initially, again with this sheep brain. We needed to show that this polymer doesn't interfere with the signals in the brain, because all our patients would return for an MRI scan after the operation, and it would be important to know that the polymer does not interfere with the brain signal and also the tumor signal. And you can see here, this is an MRI scan on the left, the CT scan on the right, and even to an untrained eye, you can see that clearly there's a region here which looks quite distinct from the normal brain signal, and that indeed is the polymer. And similarly on the CT scan, you can see here a little bit faint, but this is the polymer distinguished from the normal brain. And because many of our patients will receive radiotherapy, it was important that we tested that the radiotherapy course wouldn't disintegrate this material, because if it did, then the chemotherapy drugs would come out immediately, and we would ideally like it to deliver over a sustained period of time. We collaborated with colleagues at City Hospital in Nottingham, and you can see here on the left this very strange dummy is called an anthropomorphic phantom. So it's a dummy that's a human head and torso, many tiny holes around the body and head region. Radiotherapists use this to develop various protocols. So that was quite convenient that we could make our polymer and basically stuff it in the middle region of this head. And then we gave this dummy containing our polymer a 6-week course of radiotherapy, similar to what patients would receive. And this image here on the right, this is a very high-powered microscope showing that the material radiated compared to the material not radiated doesn't show any difference, so that you can see the individual particles here. So there was no reason to believe that radiotherapy would cause this material to break down and disintegrate. So this was all showing the clinical utility, but of course the most important thing is, can it deliver chemotherapy and does it work? So we were fortunate that what we wanted to do was work with the individual, the group that developed those gliadel wafers I showed you, because to date it's still the only drug delivery product in clinic. This gentleman here is Professor Henry Brem. He's a leading neurosurgeon in the United States, and he developed gliadel in collaboration with colleagues at the Massachusetts Institute of Technology. So fortunately he was interested in using our material to deliver chemotherapy drugs and using the animal models of brain tumors that he used to develop gliadel. So he is based at Johns Hopkins University in Baltimore in the US. So we've done a few studies over there. This is Dr. Smith, who you'll hear from twice later on today. He's our neurosurgeon, because this is a specialist technique. This requires conducting neurosurgery on the bulk tumor and then delivering our polymer containing drugs. So the experiment we did was to use the PLGA PEG paste, and we delivered 2 drugs in combination, etoposide and temozolomide. Temozolomide for high-grade glioma is the monotherapy that is used routinely. And you can see this image on the right. So this is an anesthetized mouse with the tumor, which has just been removed, and you can just about see there this white paste is the PLGA PEG containing both these chemotherapy drugs. And then just to fast forward almost a year's work, this was the result we found. So this is an overall survival curve. The survival number of days of survival is in the bottom, and the percentage of animals surviving is on the left side on the y-axis. And you can see here, so the first group, so if animals are not treated, this is a very, very aggressive tumor in the rat. You can see that within about 2 weeks the animals have to be sacrificed because of their conditions deteriorating. Importantly, we had an arm that included the current clinical care. So these are animals that have had surgery, they've had oral temozolomide, and they've had radiotherapy. And you can see that they do survive a little bit longer, but still don't do terribly well. But what was very encouraging is that the 2 groups where we had conducted surgery, delivered the paste containing temozolomide and etoposide, those animals survived to 120 days. And then when we introduced radiotherapy as well, they also survived for the same period, but the proportion of animals surviving increased. At this stage, we terminated the experiment not because the animals had any side effects whatsoever, but we wanted to study the brains. We wanted to excise the brains from the animal to show that the animals are disease-free. And this is the final study that we got the brains back to the University of Nottingham. You can see here, this is a whole brain section of the various groups. First one on the left is the group that had no treatment. So again, even to an untrained eye, you can see the brain is stained kind of pale purple, but on the top left where the arrows are showing, these are the high density of tumor cells, as you expect, because there's been no treatment. The group that mimicked the current standard of care, you see the tumors cells returning and filling in the cavity where surgery was conducted. But when you look at the 2 groups where we've given our paste and the 2 drugs, and also in combination with radiotherapy, you can see the surgical resection margin, but you can see that there is certainly no evidence from this staining that we can see any tumour cells returning into the site of the primary tumour. So as Beth alluded to, we can't get terribly excited because of this result, because we've seen so often in our field that you get this kind of efficacy where we effectively cure animals with brain tumours, and it hasn't translated very often at all to successful clinical trials. Having said that, for any technology like this is the minimal stage we have to get to. So the final slide is where we are with the project. So now we do want to take this formulation, so this is the paste delivering these two drugs, etoposide and temozolomide, and we're now at the stage of seeking funding that would conclude in our first early stage clinical trial. It would be most likely a clinical trial in adults for a number of practical purposes, but this is what we are aiming to do now. We're trying to think how the neurosurgeon will see this as a kit in theatre, how they will use it, and you can see here what we're envisaging. This will be a system where you'll have two syringes. One of them will contain the paste, the PLJ peg with the two drugs. Another syringe will contain the water, all pre-measured, and the neurosurgeon will mix this at the end of the operation, mix this, creating the paste, and then you can see here on the right, this will be applied to the resection cavity. So I can take one or two questions here. I'll stop now, and then I will hand over to Catherine. So this work, as I've alluded to, is a number of people have worked on it over the years, particularly Dr. Stuart Smith, who successfully completed all those animal studies that you've seen. Thank you.
Thank you. Catherine?
Is that what you said?
I don't know whether that works or not.
Okay. Talk my hands. Okay. Hi everybody, I'm Catherine. Thank you, Ruben, for the introduction. So today I want to talk to you, start my presentation by talking to you about plastics. Now I'm sure as soon as I say the word plastic, most of you in the audience will automatically think plastics are bad. And that's not really surprising considering all the media coverage of plastics most recently. If any of you watched Blue Planet 2, you'll have seen a little turtle with a plastic straw up its nose, and that's because of all of the excessive plastic waste that's left in our oceans. Scientists have now been able to quantify this amount by saying that for each foot of coastline there's at least 5 bags of plastic. Plastics also become more expensive to recycle, so as you can see there's a lot of reasons why people would automatically think plastics are terrible. But hopefully I'm going to turn this idea on its head slightly today. And I'm going to start by looking into what actually is a plastic. According to the Oxford English Dictionary, a plastic is defined as a synthetic material made from a wide range of organic polymers, such as polyethylene, PVC, nylon, etc., that can be moulded into shape while soft and then set into a rigid or slightly elastic form. And the area that I want to concentrate in this definition is this organic polymer. So what's a polymer? A polymer is defined as a substance which has a molecular structure built up chiefly or completely from a large number of similar units bonded together. So as polymer chemists, we would call these similar units either monomers or repeating units. So essentially a polymer is just the same repeating unit repeated over and over and joined together to form your polymeric chain or polymeric backbone. So there are actually a number of natural polymers that exist in the world around us that you may not even be aware of, or may not really come to think of. So for example, one kind of category of natural polymers are polysaccharides. And a saccharide is where the repeating unit here is the sugar. So for example, starch is a polysaccharide. So a potato is actually a natural polymer. Another example of a polysaccharide is cotton. So as you know, we find quite a lot of cotton within our clothing, so we actually wear polymers. Another category of natural polymers are polyamides. An example of one of these are enzymes. Now enzymes work within our body and within the natural environment to catalyse reactions all the time. Another example of a polyamide is protein. As you all know, we eat proteins. Proteins also exist in our bodies. And the final category of natural polymers that I'd like to draw your attention to are polynucleotides. Polynucleotides. So this means that a DNA is actually a polymer. So we're made up quite a lot of these natural polymers. So hopefully I'm trying to draw you around to the fact that some polymers are actually good and they actually make up a lot of the environment around us. Now as polymer chemists, we try and take these very sophisticated systems and mimic them in nature so that we can use these sophisticated systems, for example, in the biomedical field. So these are just to give you a flavour of some of the polymers that exist in the biomedical field. There's thousands of them. These are just a few that I came across. So for example, we have the prosthetic foot. There's a biodegradable and reusable EpiPen. There's biofix, which are inserted during surgery to encourage the regrowth of bone. There's Proflex mesh. Again, these are used during surgery to kind of like swab up blood. And finally, Glidol wafers that Rumen introduced earlier. And as you know, these are used as local drug delivery systems for the treatment of glioblastoma. So this leads on to the idea that polymers, not only they exist naturally, but we can also use them as drug delivery systems. So how can we do that? So as I said, we have our polymeric chain, and we can manipulate this chain so that we have at least two sections to it. Hydrophobic, which is shown in red, and hydrophilic, which is shown in blue. Now hydrophobic basically means water-hating, and hydrophilic means water-loving. So when we introduce these polymeric chains into a hydrophobic system, the hydrophobic area will clump together to form a core, and this will be surrounded by a blue hydrophilic outer layer. So essentially you have this nanoparticle made up of two sections, the core and then the outer layer. So if you introduce a hydrophobic drug into the system, as the polymeric chains come together to form this nanoparticle, it will encapsulate the hydrophobic drug in the center, and then it will be covered by this hydrophilic outer layer. So I've spoken about nanoparticle, and Ruben mentioned earlier micro, but it's kind of hard to visualize of how small we're actually talking. So nano, as most of you know, will be 10 to the minus 9 meters. But this means that a nanoparticle is typically the size of 1/100th of a human hair. So this is tiny, but it's still quite hard for us to actually visualize what this actually means. So I found this quite nice analogy to say that the size of a typical nanoparticle is to the size of a football, if the nanoparticle is the size of a football would be the size of the Earth. And carrying on with this football analogy, if a football is the size of a nanoparticle, how big are the brain cells that we'd be dealing with? So if a nanoparticle is the size of a football, a brain cell would be not just one, but 2 Wembley football pitches joined together. So that's the kind of size we're talking about, and I'm hoping that's made it slightly easier to visualize. So why are scientists excited about nanoparticles? Well, as we've established, they're pretty tiny, so we think that we'll be able to use these to penetrate further into either healthy tissue or tumour tissue. So the reason we'd want to penetrate into tumour tissue is to get to the centre of the tumour core, so that for then the nanocarrier to be able to release the drug and have an effect. Why we might want it to penetrate further into healthy tissue, as you know with treatments such as glioblastoma, the tumour is removed, but then you have these residual tumour cells left behind. So if the nanocarrier is able to travel through the healthy tissue, it will then be able to reach the residual tumour cells and have an effect. The polymeric carrier also acts as like a protective case around the drug, and this has two benefits. The first one is that every drug has an active half-life, so it means it's only active for a certain amount of time, and this certain amount of time can actually be quite small. So if you apply a drug directly into a natural environment, it's going to start degrading quite quickly, which means it's going to have a very short amount of time in which you can act upon. So therefore, it's not really going to be able to travel very far in its active form. So if you have this protective casing around it, it kind of protects this time bomb of the life expectancy of the drug. The second benefit is that it also protects the healthy tissue. So as you all know, chemotherapeutics or chemotherapy drugs are very toxic, but they're not necessarily selective just for tumour cells. They're also toxic to healthy cells, and that's why we see these side effects. So by putting a drug in this kind of polymer casing, we're actually kind of protecting we're also protecting the healthy cells from the drug's actions. So we believe that they'll have less toxic side effects, and therefore, because there's less toxic side effects, we'll actually be able to load more drug to have more of a potent effect when it's applied. We're also hoping that we'll be able to control the release of the drug, and I'll come on to that on the next slide. But the main reason why nanoparticle therapy is exciting is because it's not some far-fetched theory that's decades away from actually working and being in our clinics. There's actually formulations on the market as we speak, used in clinics worldwide. So as I said, we'd hopefully be able to control the release of the drug. And we can do this by comparing healthy cells and cancerous cells. Now, healthy cells and cancer cells have very different genetic makeups, but they also have different tumour environments, depicted here by these lightning bolts. So as polymer chemists, we can kind of take this information that we know about the tumour environment and try and build this into our polymeric chain right at the beginning, so that when we form the nanoparticle, we can try and control the release of the drug. So if we have our polymer chain, we can do some quite complex chemistry to add on these responsive linkers. And the idea here is that the responsive linkers, once triggered, would break apart and release the drug. After some more complex chemistry, we can add on our drugs to these systems. So what we'd hope is that when the polymeric chains with these drugs attached reach a healthy environment, nothing would happen because there's no triggers involved. But as soon as we introduce a trigger, this drug would be released because the responsive linker has broken down. So this hopefully comes full circle to Rumen's talk with the PLGA-PEG paste. And the kind of overall aim of my PhD is, rather than just in kind of putting in chemotherapeutic drugs directly into the PLGA-PEG paste, we'd actually put in these nanocarriers that are carrying the drugs for us. So if we can incorporate these nanocarriers formulated into the paste during surgery, we'd then be able to release the chemotherapeutic drug-loaded nanocarrier. It would travel through the healthy cells and not have an effect on them. And once it reached the tumour cells, because of the tumour environment, the drugs would be released and it would kill the cell. So in conclusion, I hope that I've been able to show you that not all plastics are bad, but I do feel like I have to make a disclaimer. Most plastics are bad, so please don't go throwing them in the ocean and not recycle or anything. Hopefully you've understood that polymers can be used as drug delivery systems, and you may be kind of wondering where I'm at with this project. So I've done quite a lot of the chemistry, but there's always more chemistry to be done, so that's kind of a work in progress. But I've got my first batch of formulations that I'm currently doing cell work on. And as Ruben said, we've got quite exciting collaborations with Johns Hopkins and also another one with the University of Leuven in Brussels. So for future work, we'll hopefully be able to go over there and do some in vivo studies. But that's all. Thank you very much.
Thank you. Wow, a whistle stop. A journey, something faster than a journey through drug delivery. Personally, I think the scale, for me, it's opened my eyes to I suppose it feels like the scale of opportunity here. There's a lot of exciting work going on, and also the diversity of the opportunity. There's lots of work going on in different areas. I think we've definitely got some time for questions now, if anybody's got any. Maybe we'll just start with 3, see how we're doing for time, and then take it from there, go into the coffee break. Has anybody in the room got any questions for any of this morning's speakers?
Thank you very much to the organizers and also to the speakers this morning. I've got one question for each of the 3 speakers. My name is Pat Wooters. I am the mother of a child who survived brain cancer. How difficult has it been to get research projects for trials on children with brain tumors? Because as we know, most of the research says that research and trials for kids with brain cancer, there's not been any progress. So I was happy and encouraged to hear some of the research today. But when will this get to our kids, and what have been some of your difficulties, and how can we help to lobby to change things and help?
Pediatric oncology is based upon trials-based evidence. So 50% of the children we treat for any disease, whether it's leukemia or a tumour in the arm, is based upon clinical trials evidence, and the children are involved in clinical trials. And to make a comparison with adult oncology practice, something like 10% of adult patients are included in clinical trials, but in childhood 50% are included in clinical trials. So we have built our career, our development of our specialty on a trials environment. That doesn't mean to say it isn't hard work and difficult, because we're dealing with very rare conditions. When you look at it, so 110 different types of brain tumour, you can imagine from a statistical point of view, getting enough patients of a common type to study a new drug means you have to make a global initiative, almost to study some of the rarer types, and certainly an international initiative. In the UK, there are no brain tumour trials in children run solely in the United Kingdom. They're all run with European collaboration. So the challenge is the collaboration. The success is the collaboration. We know each other across Europe. We know each other across the United States. So we actually have built bridges and developed collaborative relationships. Ruben's working with people in Baltimore. I'm working with people in Washington and in other countries. So that it's an international specialty. I think the biggest challenge is how we enthuse the drug industry to see that the children need drugs developed for them as well as the adults. And I think that's the difficulty. I don't think it's insuperable. There are many pediatric oncologists who have now moved from the clinic into the commercial environment to try and take the message for children to within the commercial environment. And I think that is a strategy that's starting to bear fruit. And I think the other thing is that what's happening in adult cancer is where we talked about the big 110 different tumour types, in each one had at least 1 or 2 molecular markers. Well, the same is true in adult practice. So what was lung cancer, small cell lung cancer has now got 10 or 15 different subtypes, And so for adult cancer, having to design drugs for the molecular target rather than for the biological, the kind of headline disease. So I think we will join forces in that initiative, in that we'll be having bio-targeted therapy for tumours which have similar targets to adult cancers, but they may well be entirely different tumours, and they're in children, so the way you give a drug to a child is different to the way you give a drug to an adult. So I think there are opportunities opening up, but it's not easy. I think the big thing that's happened in the last year is that this country has devoted tens of millions of pounds of research funding for brain tumour research, literally in the last 12 months, when it was not committing that money prior to that because of political awareness. And so I think the future is good.
Thank you both. Are there any more questions? Or is the lure of a cup of coffee too strong? I'm sure, are our speakers around over the break? Yes, I mean, I'm sure if you were to approach them directly, they'd be more than happy to talk to you if you've got any questions you want to take off air, so to speak. And I suggest, I can't see the clock, it's 10 past, we're 10 minutes behind. If we were to come back here at 25 past, is that okay with everybody? That gives us time for a quick break, and then we'll pick up with Dr. Stuart Smith. Thank you.